When we see the structure of DNA polymerase, it resembles a hand. The structural studies on DNA polymerases reveal that the DNA substrate sits in a large cleft that resembles a partially closed hand. So, DNA polymerase, the substrate of DNA polymerase is DNA, and this DNA resides in a partially closed hand. So, DNA is present here, and this polymerase is just like this. This is just like a partially closed right hand. Based on this hand analogy, the three domains are present in the DNA polymerase, and these three domains we can say these three domains as thumb, fingers, and palm. So in a DNA polymerase enzyme, these three domains are present. One is thumb, the other domain is palm, and the third domain is finger. So these fingers, thumb, and palm, all these domains play important role in the working of DNA polymerase. So here in this diagram, you can see this is the three-dimensional structure of DNA polymerase. It has three domains. One domain, this resembles with this thumb. The other domain is this domain, which resembles the palm, and the third domain is this domain, which resembles the fingers. Or in other figures, you can see this is thumb, this is palm, and these are fingers. So this DNA resides in this cleft, or in this palm. So this is the structure of DNA polymerase, and it resembles a hand. The palm domain, let us see the working of these three domains. The palm domain is composed of a beta sheet, and you know about the beta pleated sheet in the structure of proteins. So this palm domain, this is composed of a beta sheet and contains the primary elements of the catalytic site. You know, in the active site of the enzyme, there is one binding site and one catalytic site. So this palm contains the primary elements of the catalytic site of DNA polymerase. This region of DNA polymerase binds two divalent metal ions. So two divalent metal ions, either magnesium or zinc, they are present in this domain, in the palm of the DNA polymerase. One metal ion reduces the affinity of the 3' hydroxyl for its hydrogen. So the affinity at 3' end, the affinity between oxygen and hydrogen, this affinity is reduced by one metal ion, because this has to attack on the on the alpha phosphate of the incoming nucleotide. This generates a 3O oxygen that is primed for the nucleophilic attack of the alpha phosphate of the incoming DNTP. So to prime this 3' end, this metal ion works. The second metal ion coordinates with the negative charges of the beta and gamma phosphates of the DNTPs. That is, when these two phosphates or pyrophosphate it releases, this metal ion, it coordinates with the negative charges present on this pyrophosphate and stabilizes the pyrophosphate produced by joining the primer and the incoming nucleotide. When incoming nucleotide joins with the primer, this pyrophosphate is released. So this second metal ion stabilizes this pyrophosphate. In addition to its role in catalysis, the palm domain also monitors the base pairing of the most recently added nucleotides. As you know, the base pairing is very important in DNA replication. So this palm domain not only catalyzes the addition of nucleotides, rather it also monitors the base pairing of the incoming nucleotides.